Apparently, at least in Weissman's lab, there seems to be something slightly different in Sue Lindquist's lab. But in Weissman's lab, they seem to be monomer thickness. That is one protein thick, and then you just grow the filaments that way. Now, that's pretty interesting because this is one protein thick. How do you get this much broader one, right? That's thicker, right? Okay. Now, mammalian prions are definitely more complicated. First of all, the yeast prions live inside the cytosol. They don't bind to anything else. And the yeast prions, coming back to your question, the yeast prions do require a chaperone to get their prion function inside the cell. But they'll do the aggregation anyway. Interesting that the yeast prions tend to have high concentrations. One of them has high concentrations of glutamine, not unlike the polyQ diseases. But they grow inside the cell, whereas the prion protein is anchored to the membrane surface. And given that the prion is anchored to the membrane surface, there's some recent experiments which show that if you delete that anchor, then you lose the toxicity from the prion protein. You can still grow infectious prion material, but you lose the toxicity from the prion protein. So there's evidence that the yeast prion is much simpler than the human prion. But it's nonetheless nice to be able to prove, at least in one prion system, that protein only works, with the exception of the chaperone, and that, conformation indeed encodes strain of the prion. Yes, Cynthia?
I have a question. Given that mammalian prions or the function of those are
Yeah.
Given that the function of mammalian prions is not very well
Right.
known, and that the sequence of the mammalian prion is different than the yeast,
Right.
what makes them both prions? What do they have in
Oh,
common?
the generic, that's just the generic property that they can self-replicate in vivo. Yeah. And be infectious, right. Yeah. Okay, well, any other questions, or should we end for lunch, since probably everybody's headed to the shopping mall or something? Okay, thank you very much, and we'll continue. Schultz, who will be, as you can see, telling us about the molecular basis of genetic regulation, and he's from UC San Diego. So he's working with Peter Wallis. Go ahead, David. Yeah.
Okay. First, I'd like to apologize for my lack of caliber here. So, talking about gene regulation. Genetic networks. Well, nowadays with the genome project and all that kind of stuff, we know a lot of the genes and the proteins they code for, but still, like, not that much is known on how you regulate that kind of stuff. The genes that would be kind of sort of like the ingredients, and we're gonna look at the recipe. So today we're going to talk more about of the biological basis, like what people know about genetic networks in general. And tomorrow we're going to talk more about the mathematical methods that we use to study these. And then on the third one, those two are going to come together. We're going to see the specific case in bacteria, and how do you do the whole modeling of the decision making in bacteria. I don't know how much people know about this, so I'm going to start at the beginning. Some definitions. So a gene is it's a location, it's a piece of DNA that has a function. It's like a unit that's inherited from a cell. Basically has a function. It has usually has a place where RNA polymerase binds to transcribe the gene, and around there it might have many binding sites where other proteins are going to bind to regulate the expression of this gene. Then it's going to have a coding region that codes for the protein or the RNA or whatever function that you perform. And then it has a stop sign at the end. That's where the transcription stops. So RNA polymerase is this enzyme that transcribes the gene into an RNA molecule. Then this RNA molecule is read by the ribosome, and the ribosome is going to make that into proteins. Okay? Ribosome is a complex of RNA and riboproteins. So this is how the process goes. There's a gene with a promoter region. This is where this is the RNA polymerase. This is where the RNA polymerase binds. It binds here. Then it reads the DNA until it finds the stop code. This guy goes, so it binds over here, goes along the DNA, producing the RNA, finds the stop code at the end there, release, and have the RNA molecule. This process is called transcription. So here, here's the RNA molecule being produced. Then the ribosome sticks to the RNA molecule and reads it and produces proteins. So in bacteria, these two can happen at the same time. In eukaryotes, usually the RNA is transported out of the nucleus. So you have a transcription happening inside the nucleus and the translation outside of the nucleus. But in bacteria, they just go on at the same time. And that's good for us because you don't have like the time delay and other complications that you would have. So yeah, this process is called transcription. Okay. Now, just to give you an idea of the environment that you're going to find in the cell, we're going to compare the size of a bacteria, say like E. coli, to an electron, say like 10 meters by 10 meters by 30 meters. How big is stuff going to be in this scale? So, if a bacterial cell is the size of this room, the DNA would be 2 centimeters round, it would be 15 kilometers long. So, that's many laps around the room. And each base pair would be only 0.3 centimeters long. So it would be like the size of a thick rope, really long, thick rope. The ribosome would be around the size of a volleyball, and the RNA polymerase is around the size of a pill. So something like this. Typical protein would be around the size of a ping pong ball, and if it would fill up the room like gas, it would be like around 10 centimeters apart. So, and if we scale the 30-minute doubling time of a bacteria to, say, 1 year, the half-life of an mRNA molecule will be like around a month, and it would produce 1 protein per day. And the DNA would be replicated every around 16 months. It's like a year and a half almost. So the DNA replicates, the time that it takes for a whole DNA to replicate is longer than the doubling time. That means that you have many forms. Actually around like 2.7, 3 forms of replication going on at the same time, but you have exponential growth. And a protein would take like around 2 hours to diffuse across the room. So that's why you can actually use an approximation that the stuff is well mixed inside the cell. And both transcription and translation would take around 5 days each. We'll just go on. Okay. So these are actual units. The synthesis of RNA would be like around, first of all, the volume of the cell is around 10 to the minus 15 liters, which means that a nanomolar is around 1 per cell. That's so when you see usually people like to give the units in nanomolars in this case, that means like 1%. So RNA is synthesized like a few units to a few tens per minute. And it lives around 20 minutes. It's translated around like a few times per minute. And the protein, unless you have some degradation mechanism, which is useful in a lot of cases, the degradation time is pretty long. So it actually is diluted with cell division, and that's the main part of it. What you would consider like the degradation of the protein inside the cell. So if you look at these numbers, you'll find that you're going to have like a few copies of RNA, of each kind of RNA per cell. And you're going to have like around 10 to 1,000 copies of each protein per cell. So these numbers are pretty small. So there's a lot of fluctuation there. So it's a pretty noisy system. We're going to get to that later. Okay. So how do you regulate the expression of the gene? All the cells have the same DNA. You have many different kinds of cells. And they go through many different stages. So it's really important that the right genes are expressed at the right time, right? All of this parts of the process of both transcription and translation can actually be regulated in different ways. But the most important form of regulation is done by other proteins that bind to the DNA and either help recruit the RNA polymerase or it prevents the binding of RNA polymerase. So you can have both activators or repressors. Those are called transcription factors. And they don't necessarily have to be, but most of them are here in the promoter region where the RNA polymerase binds. But not necessarily. Sometimes because the DNA is very long and folds a lot, you can have regions in other parts of the DNA that are in space. They're close to the promoter region there, and it can regulate the binding of the polymerase here. And you can have many binding sites with many different transcription factors controlling each gene. So because the expression of a gene is actually regulated by proteins that are a product of other genes, and the expression of one gene is going to regulate the expression of another gene and so on. So the genome, the DNA, works at the net. And so that's what we're going to look at. So here is a depiction of a network, and it's very incomprehensible. You can see there are many different kinds of connections going on here. So we're going to look at a little part here. Now we're going to take a look at like well, different kind of properties these gene networks have. Here, this here is like what you would call a regular network, where all the neighbors are connected to each other. This kind of network, they have this property that the neighbors are all interconnected, but you would take a lot of time to travel through the network, right? Because if you're far apart, you have to go through many different connections until you reach the other side of the network. This would be a random network where it's fast to travel throughout the network, but sometimes you're going to have the neighbors are not close as far as the network goes. So the protein, the genes, they tend to work as scale-free networks here, where a few of the nodes are very well connected, and most of them not so much, but you have a few nodes that are well connected. Like in this case here, you can see that this guy here regulates a whole lot of people, right? So in this case, you have both properties that the guys that are close to each other, they're connected, and it will take no time to travel throughout the network. It's really all the nodes are within a few steps. That's what you call the small world network, right? People say that like any two people in the world, you can go from between any two people in the world with like 6 connections of people that know each other. Right? It's called small world networks. But that also depends on like a few key people that know a whole lot of people. Right? Okay. So, scale-free network. Also, this is a study where you take randomized networks, you take gene networks and compare them to random networks, and there's a few motifs that show up in gene networks that happen at a frequency that's way higher than random networks. Here's a few of them. There's the feed-forward loop, where this first protein over here regulates this last one here, stimulates this one, and also stimulates another one that stimulates this one too. Why is that good? Say that, for example, if this protein here needs both this one and this one to be activated. This system here would be robust against fluctuation. If you have like a short input, if you have a short activation of this first one, you're not going to have time to fully activate this one. So you want to activate this last one too, and you'd have to have like a long activation of the first protein, right? And that's small fluctuations with the system. This happens a lot. Also, we call it a single input module, where one protein activates a whole lot of other proteins, and usually activates itself. Why is that useful? Say there is some certain response in the cell, like a certain response mechanism that you need to transcribe a whole lot of different genes for a certain response. Usually that has a master regulator, which would be this guy over here. That if this guy crosses a certain threshold, it'll self-activate, and then it's going to go to really high levels and express all these guys. So all these guys can be expressed at the same time, and that's the advantage of having this one regulator in the same proportion too. So this is good in systems where you need these guys to be expressed in the same proportion. Also, with this, you can also have, if this guy's, the strength of the regulation of these guys over here doesn't have to be the same. So you can actually have some timed response to, say, like if this guy here is activated with smaller levels of the master activator than this guy here, is going to be activated first, right? And as the level of the master activator keeps going up, you get to activate other guys. So this happens a lot. And also you find this regions that are really densely connected, where a lot of different transcription factors activate a whole lot of other genes, all with different strengths. And that's where the computation of the network goes on, right? Because each of these guys is going to respond to differently to a different configuration of all these other regulators over here. Okay, so yeah, so these are all resources that gene networks use a lot. Now here's a pretty controversial subject: modularity. Can you take a network and divide it into functional pieces that interact with each other? Like, this is another way to depict a network, and you can try to isolate separate parts of the network that would design that would perform a different a specific function. And so a function will be performed by that module over there, and then that module will interact with other modules. There's a lot of that would make things a lot easier, but that's usually not always how it works, right? Sometimes everything is connected, and you can't really make the separation. But there's some interesting studies along these lines. So in this work over here, you get a network. This are all NOT and logic gates, and you use a genetic algorithm kind of stuff to make this network evolve. Each step you're allowed to create other gates or create other connections, and then you have a fitness function that's going to test this network and see how well it scores, and then you keep the good ones and discard the bad ones on each generation. And you have this 4 inputs over here and 1 output over here. And basically what you want your network to do is this X XOR Y and W XOR Z, right? Then here is how well your network is scoring according to this fitness function. How well it's performing this, and these are how many generations it takes, right? So you can see that if you in this scenario, your network is going to evolve to something that it wouldn't be at all like what you would design yourself. It evolves to something totally interconnected, and you can't really separate this into this function you're using here, right? Now, if instead of having just one fixed goal, like in this case, you have a goal that changes every 20 generations in this case, I think. So if you change your goal to between these two over here, I think the two goals over here, one is this one, and another one that has an OR over here in the middle. So your fitness function is going to be scoring that network. 20 generations in one of the goals, and 20 generations in the other one of the goals, so on. Now you can see that it takes more time to score high in the fitness function. But then when it finally does, the network evolves to something that can switch really easily from one goal to the other. Here's a small piece of this end here, and you can see that whenever the goal changes, it really quickly adapts to that new goal. And in this case, then here you see what you can translate as here you have the equivalent of the logical functions that they're actually performing, right? And you can switch from one to the other just with these two mutations over here, okay? And now you can see the network has modules that will perform in front of the logic functions that you initially wanted to. Okay, so basically what this tells us sometimes when you have to respond to different conditions in your environment, you're gonna have like whole blocks within the network that are useful for some specific region, for some specific reason that they can be like turned on or off, and those are going to be in the module, right? But that's like only if they have to be turned on and off. So you have a lot of that if you're working with like a rich environment where you would have to change your goals for survival often. But if there's something that if there's some set of genes that are always going to have to be on, they're eventually going to merge into something more interconnected and hard to break apart. Okay. Now, noise in gene networks. That's actually what we actually work with. Usually, when people are talking about the networks, people usually nowadays use deterministic methods. They're definitely very useful. But nowadays, there's a lot of discussion if noise is just something small that you're trying to prevent in a network, or if it's actually something that's useful and it will help the cell make decisions. So usually people use these deterministic methods and they add some logical type noise to the equations. But due to the discreteness of these concentrations, more and more people have been using stochastic methods. That's what we've been doing to deal with noise. So this is an experiment where you put two identical promoters, one of them expressing this green fluorescent protein. Oh, it's actually not green and red, but in the picture it shows green and red. I think it's like cyan and yellow. But anyways, one goes for this green protein, and the other one expresses this red protein. So the promoters are identical, right? And they're within the same. They're inside the same cell, so they should be exposed to the same conditions. Okay. Now, if the noise would come from the environment inside the cell, these two genes should have different. Their expression is not going to be synchronized, right? So, if their expression is synchronized, that would mean that the noise comes from the outside and not from inside the network. Right? And if their expression is not synchronized, that means that comes from noise in the expression problem, and we call those intrinsic noise and extrinsic noise. Okay. So you have, in this case over here, where the genes are being fully expressed, they're being expressed a whole lot. You see that the fluorescence level is the same for both colors. That means that because they're being expressed like so much, there's very little noise going on inside the process of expressing the gene, right? And all this fluctuation that you see here is from other parts of the cell, other components of the cell. And that's why both of those genes are exposed to the same environment, so they have the same fluctuations and the signals correlate. Now, if you have the genes, in this case over here, you have the genes being regulated. So you have repression from other transcription factors inside the cell. So in this case, you can see that the fluorescence signal doesn't really correlate at all. So all this difference here, all this noise, comes from inside comes from the transcription and translation of those genes themselves. So that's why you see cells with all sorts of different colors here. You see red ones, green ones, yellow ones. And here you have a plot of two different strains of this bacteria. I think it's E. coli or something. This is a red fluorescence and this is green fluorescence. So anything along this axis over here is any spread along the diagonal would be extrinsic noise, okay? And anything this direction over here would be intrinsic noise. And you can see that the two cells and the two strains can have different characteristics. The green one has a lot more noise than the blue one. So it really depends on the case, but yeah, you have both kinds of noise and they're both important. So this is like fun stuff that people do with this kind of thing. This is the repressor layer, and I tried to find the movie for this because it's really cool, but I couldn't find the movie, so we're gonna have to be happy with this picture here that you can barely see. Here you have three. This is a plasmid, which is a small piece of DNA, circular piece of DNA that people can insert into bacteria, and it's going to express whatever you put in there. So here you have three different kinds of genes that they repress each other in sequence here, right? We have gene 1 repressed gene 2, gene 2 repressed gene 3, gene 3 repressed gene 1. So if you took the parameters right, and one of the genes expressed is a green fluorescent protein that you can see the fluorescence, right? So if you took the parameters right, you can get a oscillating behavior in this network over here. So here's the fluorescence in time. You can see how it oscillates beautifully here. And here it's hard to see the cell, but you can see that it's dark and light and dark and light and dark. And interesting, the period of this oscillation is a lot longer than the doubling time of the cell. Yeah? So you can see if I had the movement, you can see the whole column of the cells, like they're moving around and dividing and doing their thing there. And every once in a while, the whole column is going to be bright green, and then it's going to turn back off. Then now, another about this lambdaphage, which is a virus that it's a bacteriophage. So it's a virus that attacks bacteria. This one, the lambdaphage, that attacks E. coli. And this is a really well studied case, and there's a lot of work done on this, and it's like
The hydrogen atom of gene networks.
as they say, it's the hydrogen atom of gene networks. Basically, the way they found out about this virus is you get a colony of E. coli, right? And then you shine UV light in this colony, and that causes like a bunch of the cells to start producing this virus, and the virus is going to replicate a lot, and it's going to attack the whole cell, and eventually the cell is going to lyse and release all this virus in the media, right? This is what you see over here. This is an infected cell. So the cell's like living there, no problem. Then you shine UV light, and this happens. All the little viruses explode the cell and get released. There's a picture of the virus. So, okay, what happens there? This virus, it has it can do two things. It can either incorporate its DNA to the DNA of the bacteria and just live there happily, and the bacteria keeps dividing, and when the bacteria divides, it replicates on DNA and divides, and so the DNA of this virus is also replicated and divided into the cell, and the cells are there, no problem, right? While the cell is happy, the virus is happy there, and the virus doesn't do anything. Whenever there's any kind of cell stress, in this case the UV light, but it can be all sorts of other kinds of stress in the cell, then the virus is like, oh, there's something wrong here. Then it takes this other pathway where it replicates itself a lot and kills the cell, right? So here's a virus-free cell. Virus-free cell gets infected. Now here's where the decision is made, right? This virus can either incorporate the DNA to the cell and live like that, right? Or it can lyse, or it can replicate itself and lyse the cell, and then all these viruses go on to infect other cells. Okay, now, how is that decision made? Here's the DNA of the virus. You have this genes over here, these lysogenic genes. So when the virus is not killing the cell, when it's just passing its DNA along with the cell's DNA, that's called lysogeny. And that's why you express these genes over here, the lysogenic genes. When the cell takes the lytic pathway, there's all these genes over here, early lytic genes over here, lytic genes over here, and those are the genes that are going to make copies, many copies of the virus and assemble the virus and kill the cell. And the whole decision is made over here. These are genes right here, where you're going to decide if you're going to go this way and transcribe these genes, or if you're going to go that way and transcribe those genes. Okay. Here's a close review of that. So here, the CI over here is where you go transcribe the lysogenic genes, and the crow over there is where you're going to transcribe the lytic genes. These are two regions where the RNA polymerase can bind. And overlapping with those regions, you have this binding sites over here, they're called OR1, 2, and 3, where both these transcription factors over here, CI, which is the transcription factor involved with the lysogeny, and CRO, which is the transcription factor involved with the lysis, they can both bind to all three of these sites with different affinities, and regulate the binding of the RNA polymerase to the promoter. Okay, the CI transcription factor, the one involved in lysogeny. It's a dimer like this, and it has these two domains over here. One of these domains binds to the DNA. It's good for binding to the DNA. And the other domain is good to binding to each other. Okay? So that's why they form dimers, and that's why this what happens. It has a really high affinity to the first binding site, and then like a 10-fold less affinity to the other two binding sites, the 2 and 3. So at a pretty small concentrations, it can bind to the first binding site, and when it does bind to the first binding site, it has this it recruits another dimer to bind to the second binding site. So a pretty low concentration. You have the first one bound, and then you recruit another one for the second. And when that happens, these two guys over here, they recruit their RNA polymerase to bind to the promoter over here. And the thing is, the binding affinity of the RNA polymerase to the promoter here is otherwise low. So you need these two guys over here to recruit the RNA polymerase to bind over here. Okay. Then eventually, if the concentration of the CI is way high, then you have all three sites bound, and you won't have any binding of the RNA polymerase. Okay, so what happens here? If you remember, the other binding site, the one that will take the cell into lysis, is over here, right? So if you have these guys bound here, the RNA polymerase is not going to be able to bind to this region over here. So you shut down the lysis, and you recruit the RNA polymerase to express these genes over here, and among these genes over here, the coding for the CI is also there. So these two guys bind over here, they transcribe these genes, so more of these guys are produced, and you shut down the lysis. Okay, so that's how the cell, that's how the virus prevents the cell from going into the lysis pathway, and it lives happy like this. Now, if there's any kind of cell stress, one of the responses that the cell has, there's this enzyme that that's involved in like cleaving some proteins around the cell, and this enzyme can also destroy these guys. So under cell stress, these guys are degraded fast. So what happens is you have this you have like again like all the sites free, all the binding sites free, and then what happens then is the RNA polymerase is free to bind to this promoter over here and transcribe the lytic genes. When it does that, it starts transcribing the lytic genes, and among these genes there's this CRO transcription factor, which is the one associated with the lytic. Okay, is this guy over here. They also bind like a dimer, but they don't recruit each other or anything. And their affinity for the binding sites is the opposite of the CI. It binds more strongly to the OR3 binding site and less to the other two. So then, the RNA polymerase starts transcribing these genes, produces CRO, and CRO will first bind to the third binding site over here. What does that mean? That prevents the RNA polymerase from binding to the CI promoter. So that shuts down the lysogeny pathway and leaves the possibility open here for transcribing of the lytic genes. And from there, all the lytic genes start being transcribed, and then the cell goes into lysis. Okay. That's how the decision is made. Now, just to finish for today, I'm just gonna start showing to you guys how do you usually model this kind of networks in a deterministic way. This is usually what people do. So you write down this kind of kinetic rate equations, okay, where you have a basal expression rate and a fully induced expression rate that's modulated by this Hill function over here. And it's written like this. But in the case of regression,
Will the boarding will work each of these terms? Work each of these terms. What's alpha 0, alpha 1, for the unit say that?
okay.
This work is good, but the way I write the equation for
Okay, so I have a protein A, I can do A by B. Okay. Then I can just write a differential equation for huh? Okay. So DADT. You're going to have the basal expression rate, and that's always going to be there, and that's usually small. Okay, and then you're going to have on top of that the like a fully induced rate, and that's going to be modulated by a Hill function over here. Now, this so
Vamos ter que fazer.
okay. So these Hill functions are functions that go from in case of regression, they're going to go from 1 to 0, and they're going to switch at some equilibrium value here. And in case of activation is, they're going to go from 0 to 1. And then how sharp this transition is going to be, it's going to be determined by the Hill coefficient here, this n over here. So if you have a high coefficient, you're going to have this transition being really sharp here. If you have a low one, it's going to be something really slow like this. Okay? And that is what you get with cooperativity. If you have, for example, if you have the protein binding as a monomer to the DNA, you're going to have this n equal to 1. And if you have the protein binding as a dimer, you're going to have something with the other two. And so on. If you have like bigger oligomers, you have higher, like sharper responses. And then you have a degradation term over here. And the degradation term is proportional to the protein concentration. This thing. So the question is something like that. So if you have like we're gonna see tomorrow, more like a stochastic view of the system, where you consider the binding and unbinding, the synthesis and degradation of the proteins as stochastic events. This deterministic approach here would correspond to averaging the binding and unbinding. Just put an average synthesis, and it's weighed by. So here, if H is the binding and F is the unbinding, H over H plus F is the amount of time that the cell would be bound, and F over H plus F is the amount of time that the cell would be unbound. So then it would average the bound and unbound synthesis rates over there. You get something like this. So sometimes, in most cases where the binding and unbinding is really fast, that's okay to use this, but not always. So now, just to finish here, this is the kind of stuff you have. This is a little system of two genes that they repress each other. Okay? So gene A represses gene B, gene B represses gene A. So if you write down the equations, in this case for the binding of monomers, you're going to see that the repression here is the Hill coefficient is going to be 1, right? Now, you got these equations, and here there's a plot of the Milk lines, which is the dAdt equal to 0 curve, and the dBdt equal to 0 curve, right? And in this case, where you have binding monomers, you're only going to have one intersection, only one fixed point. You're going to have one stable fixed point. So your system is going to be around this point over here, right? Where the lines intersect. This is just a plot of these milk lines at this point, and this is the flow. Okay, I can see that all the arrows point over here. Now, if you have a cooperativity, if you have a binding dimers, here, like then the binding term over here is going to have the Hill coefficient of the other 2, right? Now you can have if you plot the same two milk lines, you can find two stable fixed points, and you have a bistable system. You have a switch, where you can either be in a conformation where there's a lot of protein A, and you shut down the production of protein B, and therefore the repression of protein A, or the other way around. Yeah. So, well, nowadays there's a lot of that's something we're working on right now too. Is what do you actually need to have this bistable system? Do you really have to have cooperativity, or do you have to have noise, or do you have to have a stochastic approach or not? But yeah, this is usually what people have been doing with this deterministic view. Yeah, I guess this is good for today. There's any question?
So then, how would you get the bistable system and compare to the lambda phase, the lambda switch?
Well, there's a lot of papers with simulations on the case of the lambda switch, and the thing is that the lambda phage is a little bit more complicated than what I just put over there. There's some other kinds of regulation from other parts of the DNA, and the degree of cooperativity there is a little bit more complicated than what we saw over there. But there's been some successful simulations of the lambda phage, but they include elements that I didn't show over here.
Yes.
Yeah.
In the beginning of your talk, you showed some slides where you showed nodes and things like this. how do you know, how do the people discover these nodes? By microscopes or by other techniques?
You can, well, there's many techniques that biology use. Huh?
It's like that.
Okay.
It's always good. Refresh people's memory.
So many things you can do. You can, for example, mutate one of the proteins. So you have you delete one of the proteins and see and see what happens downstream. See if you don't have that protein there anymore, that's going to change the concentration of which other proteins, right? So then you can see if that protein, if any protein is affected by the presence of the protein you deleted or not, right? And you can see if the concentration increases or decreases, and so on. But that doesn't necessarily mean that protein is going to regulate those proteins directly, right? You just know that it's downstream somewhere, but you don't know if that protein binds over there itself. What you can do for that is there's like microarray experiments where you have all, for bacteria you have the whole genome of all pieces of DNA bound to a glass plate, and then you can put proteins over there or anything else over there and wash it, and then see what sticks where, and so you find out about binding sites and proteins, different proteins that bind to different parts of the DNA and like that. There's other techniques too, but I'm not even experimental, much less a biologist.
But that just to make a comment here that I think questions interest for people to think. Most of these techniques, like gene arrays that people call sort of whole genome techniques, they're very good techniques to sort of get the sort of average view, but you lose a lot of spatial information, time information. So you know that something binds. So and a lot of things also when you knock out the gene and you don't have a protein and you go down, you also lose these things out. One of the things that people are doing these days is actually people are becoming very, very good about creating techniques where you can do time domain of these things, like creating fluorescence probes that go there, do single cell experiments. There are situations, particularly bacteria, where people are learning, for example, to put one gene and with a single promoter, you can put that gene and GFP gene next to it. So every time that gene gets activated, GFP gets activated. So you start to follow these things around. But this is sort of the real state of the art. So on one end, you have all these high throughput techniques that you figure out these things, but you lose all this sort of time information. You have just this average field, right? It's just like if I want to see how many parts you have, I completely destroy you, and I count one eye here and a hand there, but I don't know what exactly all these things stick together. That's what this whole sort of whole genome techniques do. But it's sort of new probes and optical probes and things like that people are doing. So I don't know how much time we're going to go through today. We're going here. We don't have a real experimentalist doing that. We're going here. I think this idea of what people call single cell experiments is the way to the future. And there's a lot of interesting physics that goes in there. I think you're still about the theoretical tools to do that, but
there's a lot of experiments done with this fluorescent proteins. I don't know if you know about this. It's worth commenting. This green fluorescent protein is a protein that they get from what do you call it? Jellyfish. That's fluorescent. Yeah, if you shine light on it, it's fluorescent. So there's a gene that codes for that protein that you can insert in any part of the DNA that you would want to study. You insert that gene over there. So say there's a certain protein A that you want to see the pattern of expression of that protein. You just attach before the stop codon. You just attach the code for this fluorescent protein. So every time that the protein A is expressed, you'll also express the fluorescent protein. So then if you measure the fluorescence, you can measure the level of the fluorescent protein, and you know that that's the same level as the protein in the Chinese study. So that's basically what it is, but there's all sorts of different experiments that you can do with that.
Yeah, just to complement on what Nelson said, I mean, so that means that we are going back to biology, because for a while people thought that once you had the genome, everything would be solved, but that's not true, because you depend a lot on protein-protein interactions, well, homomeric interaction, heteromeric interactions, and especially when you think about interactions with nucleic acids, I mean, both DNA and RNA, of course, the Ebola is a much simpler system than eukaryotic cell. But I think, like you showed, I mean, how important it is, and I think it's correlated a little bit to the Daniel talk. I mean, why proteins, they like to bind one to the other? I mean, and in principle, because sometimes it is functioning in a different way. So all this protein, all this binding, all these preps or activate, usually they are dimers. But in some circumstances, they make tetramers, or prime tetramers. And sometimes it does depend, I think, the case of the lambda phage repressor acts in another place. And I'm sure that it will be the same for the probe, because also shows for XA repressor. It can make tetramers depending on the pH and the DNA is also not linear, because it might be curved. I mean, because of 53, for example,
yeah, that's the first slide. They actually moved the first slide, etc. On that basis. Let's comment on this sort of they control that basis.
So, and then you have all sorts of possibilities, and more recently, a lot of people, including us, have found that a lot of these DNA binding proteins, they tend to aggregate just because they in their function they have to connect to each other, and so, and then I think, I mean, if you take, I mean, this time domain here, that's quite complicated, but it can be assessed by theory, that's great, but also, I mean, only experiments, I mean, I think that need to go in